Hello and a warm welcome to our latest edition of our current webinar series. Today we have the great pleasure in presenting to you in vivo pharmacokinetic experiments in preclinical drug development. I'm Sascha Hartmann, Business Development Director at Samiris, and I've got the great pleasure to introducing today's speaker to you. Dr. Janne Mannila, Head of in vivo pharmacokinetics at Admescope, a company part of the Samiris family. Janne has a background in pharmaceutical sciences and has more than 10 years experience working as a senior scientist in CROs in Australia and Finland. He has in-depth knowledge of in vivo and in situ pharmacokinetics and preclinical formulation development. Janne established the in vivo laboratory at Admescope on his joining the company in 2018. He's contributed to more than 15 peer-reviewed research papers, book chapters, and international patents. Of course, this webinar is interactive, and we encourage you, the audience, to participate by submitting questions. You can submit your questions using the live chat tool in the application. And Janne will do his best to address your questions at the end of the talk, time permitting. But don't worry, if you don't get a chance and think of a question later on, you can always submit your question directly to Janne by email or into our contact email box at Samiris, or contact us via LinkedIn, and we will get back to you. Before we dive in, I'd just like to remind you or encourage you to follow Samiris and Admescope on LinkedIn to keep up to date with future webinars and details on our service offering in drug discovery and development. But now, without further ado, over to Janne.
Thanks, Sascha. It's a pleasure to be here, and good morning, good evening, wherever you are. So the topic, as Sascha said, is a in vivo pharmacokinetic experiment in preclinical drug development. So I'll be focusing on in vivo. I'll be focusing on the in vivo stuff first, mostly, but also in some of the areas that are closely related to these experiments. Talk a little bit about Admescope. So Admescope was found in 2011, so it just turned 10 years. We are an ADME tox CRO and specialized in drug metabolism, drug interactions, pharmacokinetics, and bioanalysis. And where I'm based, Oulu, is the site of our headquarters and the main research site, including the animal or the in vivo lab. Here are some images. Well, firstly, Oulu is here up in the north, and as you can see, it gets cold here, but we also have nice summers. So we get all the seasons. And I should say that Admescope started as a company that was built strongly around the knowledge and in metabolism and mass spectrometry. So that's our main core. Sascha also mentioned, I actually joined the company in 2015, but the in vivo lab was established in 2018. And by establishing the in vivo lab, I mean we built the new lab in our premises. Before that, in 2016, we started in vivo work at the university where we had our private room hired. And when we build our new facilities here, at the same building where we have our offices and other research laboratories, it was tailor-made for the animal work. Let's then just briefly the services we provide. So in vitro metabolism, drug-drug interaction, in vivo DMPK, quantitative bioanalysis, permeability and transporter assessment, physicochemistry and binding, in vitro tox, and biologics, which falls under many of these previous categories as well. But if you're interested in any of these, then get in touch and I'll introduce you to the relevant head of that particular department. All right, so just briefly what I'm going to be talking about. I'll be first explaining why the animal research is needed and the legislation, but that's only going to be a couple of slides. Then I'll go through the a typical in vivo experiment. Then I'll just briefly touch bioanalysis and preclinical formulation developments. So firstly, why animal studies are necessary. We always want to, of course, when we have new compounds, we want to test them quickly and to inform the further synthesis of new compounds. So most tests early on are in vitro tests, and of course, these days also in silico plays a role. But at some point, it's necessary to see how the compound behaves in a specific tissue or in the whole body, and that's when the animals come in. The body of an animal, or more so human, is very complex, and there are a number of systems and everything's interrelated. So it's, even though we have cell lines for absorption and so on, and metabolism, we can use them, and we do use them. It's still hard to combine all those, or it's actually impossible to combine all those. So that's the reason to use animal studies. And as you can imagine, not everyone can just start running animal experiments, but it's governed by EU legislation. And also in Finland, we need to apply for a permit from this National Animal Experiment Board. This is the case especially in smaller companies as Admescope, compared to some of the large pharma where they might have their own animal experiment board. But I think this is a good transparent system. Many have heard of 3 R's. So 3 R's are replacement, reduction, refinement. By replacement, we mean that any experiments that do not involve animals should be run first, and if we get the answers that we need, then we're happy. But if we don't get the answers, then only at that point we go to animals. So basically that's the absorption, the cell lines, cell membranes, liver, hepatocytes, and so on. So that's replacement. Reduction is the basically number of animals in this instance. So we want to make sure that we get the answers that we want by using as few animals as needed, and also by using some techniques that are new, recently become available. For example, micro sampling, and also the instrumentation, the mass spectrometers have become better and better, so they allow for smaller and smaller samples to be analyzed. We use all of those methods, of course, as well. Refinement then tells us that we should, of course, treat the animals as best as we can and give them minimize the pain, suffering, distress, and that's also described in the animal ethics application, of course. And you need to give them places to hide, for example, some bedding materials that they like to dig and burrow. But I'll show some photos a little bit later. We have sought for this accreditation from an independent organization called ALAC. So that's basically in Europe, people in many instances know that the EU legislation governs us anyway, but just to explain the quality that we have sought this accreditation from Association for Assessment and Accreditation of Laboratory Animal Care. So this is an independent accreditation. All right, let's then move on to the animal, or in this case, I've got a slide that depicts the in vivo system. And this is, of course, quite simplified system. So here I've got administration, and when this means the administration of the compounds. So we can administer to intestine, we can administer to blood circulation. If we inject to intestine, we can see how the compound moves to the blood circulation, and this process is absorption. Then if we study the distribution of compounds from blood circulation to tissue, then this process is distribution. Here I've got boxes for tissue and brain. Of course, brain is a tissue, but also very commonly there's a common interest towards brain, so that's why it's separate. Then next comes metabolism in this image anyway. So metabolism is the breaking down of the compound, and usually that happens in the liver, and liver is of course the organ that's designed to metabolize. And then lastly, we have excretion. So excretion of either the intact parent compound or metabolites can happen to urine or to feces. And we can collect samples from each of these processes, and then by combining this information, we get an idea of what's going on in the body. So then the actual requisites for running an experiment some of them are described here. So when we get an order for an animal study, we of course plan the study with the customer. We then, after we are happy with the details, we order the animals in from a commercial supplier, and the animals arrive to Admescope where we put them in these boxes that are called IVC. I'll explain that shortly, but we have about 60 of these boxes around at Admescope. These boxes are provided with bedding material and all kind of nesting material that the animals like to use, cabins and cylinders for them to play. And they are quite active, these animals. We let them just live there for 5 days before starting the study, and that's considered sort of a standard acclimatization period. These boxes are called IVC boxes, which means individually ventilated cages. And the air within these cages does not mix with the room air the workers breathe, but it's being filtered by a separate unit, and also the separate unit takes care of the temperature that's very strictly governed, and the humidity, the air exchange rate is also, they are important. And apart from that, there's the light cycle that needs to be 12 hours of light, 12 hours of dark. The animals are given access to food and water at all times, and of course we can remove the food if needed for a particular experiment, but that happens on the previous night, basically before the experiment, and we need to check the animals daily. So the next slide, I'll just show what the cage looks like. The airflow is just depicted there, and it's so it comes from the rack that holds the boxes, and then it goes back to the rack after it's circulated around the IVC cage, and the animals can be there just next to each other, even different species, because they are not able to see through that plastic. Here are some photos of just animals enjoying a nap or the refinement we've given them. Okay, then of course, next comes dosing. For dosing, we of course need to prepare the compound into a formulation that's almost in all instances, it's some sort of a solution or suspension, but liquid anyway. So we for example, for oral dosing, we grab the animal like in those pictures, and we with a very firm grip, and then we just insert this instrument, which is just syringe basically, and this is a gavage needle into the stomach. And for IV injection, we need to restrain the animals in these restrainers that are designed for the purpose, and we have different types of needles depending on the species. But for example, we often use these insulin needles for dosing. Some other relevant information about dosing. For example, the compound requirement, even 5 milligrams of compound could be enough for a small mouse study. Of course, this is very important thing to consider when at this point there's normally not that much compound around. And then we can together check whatever the route of administration should be. We do that in collaboration with the customer. Of course, we provide help if there are questions. And some of the dose volumes are listed here. So basically, the common ones would include bolus injection. So that's the IV injection, and we would ideally want to inject 2 milliliters per kilogram, and that's for a typical mouse that would be 60 microliters. And for oral dosing, we would ideally want to give them around 150 microliters. These volumes are also fairly accurate. One thing to consider is the syringes that we use, the increments, the lines on the side of the syringe, they dictate the accuracy of the dosing, and if we try to inject them, say 10 microliters, that wouldn't be very accurate, unfortunately. All right, so after we've dosed the animals, we will start collecting samples. And actually, blood sample collection is the basis for pharmacokinetics, because that's in the definition of that process. When people talk about collecting blood samples, plasma samples, they usually, of course, we need to collect blood first, but usually plasma is then stored. And that's also the basis for pharmacokinetics. So we need to separate the blood. The volume of blood that can be collected is of course very important. So typically the volumes are from 10 to 120 microliters depending on the species and the study design, how many samples and so on. For example, from a rat you can collect 8, 10, even more samples over 24 hours, and these samples can be more than 100 microliters, but from a mouse you can either collect 3 or 4 samples that are maybe 30 or 40 microliters, or you can collect more samples that are 10 microliters. The samples are collected by puncturing the vein, and usually the hind leg, or we can collect from the heart under terminal anesthesia. In almost all cases, the blood is collected into EDTA, so can't remember the compound name, but that's the anticoagulant that is used quite typically, but we can use other anticoagulants as well. And if the compounds very labile, we can even add some additives, so say acids are used commonly, or protease inhibitors in some instances. We can add them to the vial where the blood is collected. After that, the blood is centrifuged for plasma, and that plasma is then stored in freezer or deep freezer. And these, I have just included some quite typical time points. So if a customer would just come and say, we would like to run a PK study, I would most likely offer these time points. So for an IV injection, we need to have more sample times around the first 30 minutes, and then for an oral administration, we want to have samples around sort of around 1 to 4 hours, maybe, just to try and capture the maximum concentration in plasma. And we can collect samples depending on the half-life of the compound, even up to, so quite typically up to 48 or 72 hours, but even up to 4 weeks is quite common. 4 weeks for antibodies. Collecting the blood samples, the rule that dictates the collection is that the volume cannot exceed 20% of the total blood volume of the animal in question. I think these I explained earlier, but 8 to 12 samples from a rat, 1 to 3 samples from a mouse, but if we use these microsampling techniques, we can collect more samples, and these have become more and more popular as well. Then just the techniques that are used for blood sampling. Venipuncture means puncturing the vein, so we can you do that with a needle and then just allow the blood to drip. We can collect it into a specifically designed capillary tube, or we can use safety lancets and so on and so forth. These are all different types of techniques. Just I think one important piece of knowledge is the microsampling is used more and more, and that microsampling means collecting about 10 microliter samples. So that saves animal numbers quite a bit. And here's just a picture of blood collection. So there's a tiny drop of blood in the hind leg. It's been pierced with a needle already. And now we are collecting the blood using this capillary, and the capillary is preloaded with some anticoagulant to be able to then centrifuge or to push the blood out. So basically we have these types of small syringe looking equipment for collecting small samples. Also we can collect full blood into this absorbent material, and this is called mitra, or we can just use these traditional vials. All right, so then tissue sampling, we can collect any tissue. Normally, before collecting the tissue, we want to make sure that the animal's free from any remaining blood, so we rinse the blood circulation with saline. That saline can be refrigerated or whatever temperature. Some people like to use 37-degree saline. That's up to them. We tend to use refrigerated. This only takes a few seconds, or 10 to 30 seconds, but I mean, you need to balance. Of course, in theory, the compound could flush out as well. And in the next slide, I have just listed some typical tissue types that we collect quite often. So as I said, brain's probably the most important, and it's, I think, probably in 90% of cases we collect the brain, and then other tissues, and even some rare ones listed there, like bone marrow. It's not an easy tissue to collect, but we have done those. Usually the tissue can then be frozen if needed, either dry ice, liquid nitrogen, or so on. And for analysis, the tissue needs to be homogenized. We use this type of bead homogenizer that's rotating with the vials with small ceramic beads that breaks the tissue very rapidly in only a few seconds, so there's no heating of the tissue that much, but there are the other systems available as well. And then the tissue concentration that we measure shortly, we compare that to the plasma concentration at that same time, and that's an important value to consider. Then I've got a few slides of excretion. So for studying the urinary or fecal excretion, we use metabolic cages like this. Looks like a piece of work, but the animal's basically here on this metal grid, and then when the animal urinates, the urine flows into this cylinder, and same happens for the droppings. They go to a separate cylinder. If we have rats housed in these cages, there's only room for one rat here, but for a mouse, we can put a group of mice there. These samples are then pooled, so we, for example, collect them from 4 to 8 hours and maybe 8 to 24 hours. We just need to weigh how much urine or feces have been expelled during that time, and then the samples are analyzed. So feces need to be homogenized, and urine is much quicker to analyze. Another few photos of the metabolic cage with the rat there by himself. All right, so let's now move on to bioanalysis. After we have collected all the samples, we need to analyze them to see how much compound is present. And the instruments that we use typically consist of LC pump and column, and then the mass spectrometer. And we have few types of mass spectrometers around, but we at Admescope we typically use this LC-MS system. Of course, there are other types of analytical instruments available as well. But before analyzing the samples, they need to be purified so that they are no, say, proteins that will block the instrument or crash out. So we the most simple approach is to precipitate the proteins by adding just organic solvent to the plasma and then centrifuge the plasma and then analyze the supernatant. That's pretty straightforward and usually gives a good recovery. If it works, then we will most likely use that method. If it doesn't work or customer has specified a limit of a sensitivity that they would like to have for the experiment, then we can try some other purification techniques as well. And these include, for example, plates that are designed to remove phospholipids from the matrix, and also we have solid phase extraction plates and other types of tools as well. But they are more and more laborious as we go down the list here. So if a simple method works, of course, that's then used. Generally, for bioanalysis, we want to have the method selective and sensitive. So that means that we want to be sure that we are seeing the compound of interest, and also we want to see really low concentrations in plasma or the matrix. And as I've said a couple of times, at Admescope, basically the company is built around the strong knowledge in mass spectrometry, and these are the instruments that we normally use. First, after the samples injected into the system, it's separated in the column, and then it goes to the mass detector with that. If it's equipped with the triple quadrupole mass spectrometer, we can split or break the analyte into daughter ions, and then that we know that if the parent or the active compound, for example, breaks to a specific ion, then it's most likely that compound. Of course, we make sure we have the analytical standards as well, but sometimes there could be many other compounds with the same molecular mass, but it's not common to have another impurity or whatever compound that's got the same mass and the same daughter ion. So that's why it increases the selectivity of the method. Due to this, the run times are fairly short, so maybe 3, 4, 5 minutes just for running one sample. But given the number of samples, which could be 100 or 200, the analytical run often takes overnight, over 2 nights or so on. Something that we always check is this phenomena called matrix effect. That means that the matrix, whatever that is, tissue or plasma, they may have some endogenous compounds that may interfere with the analytical process, but we do check that here at Admescope just to remove that possibility. And lastly, the detection limit for small drug-like molecule is usually from 1 picogram per mil to, say, 10 nanograms per mil. So that's usually where it's at. This is the last slide for bioanalysis. I would just like to mention that in addition to triple quadrupole, we have high resolution mass spectrometers, which have become a great option during the last 10 years. So basically they offer almost the same sensitivity, but they also offer the possibility to store the data and then go back to the data afterwards and scan for metabolites even if we don't know them before running the samples, we can check later for metabolites. This is the results then. So after we've run the analysis, this is something that we might get. So for example, in this case, we have run a study for compound 1 in rats and dosed them intravenously at 3 milligrams per kilogram and orally at 10 milligrams per kilogram. And the plasma concentrations are presented here. If we then look at the graphs for the same data, this is something that we see. This is linear scale and this is the semi-log scale. And furthermore, if we check the mean to standard deviation plus minus standard deviation graphs, this is something that we see. This data then can then be the numeric data can then be used for calculating the PK parameters. I will just here present the results for this data, which are calculated using Win-on-Lin. So basically, some values that are of interest here are, for example, half-life. AUC is the area under the curve, clearance, and volume of distribution. I'll explain them shortly. And we have the same results for the peroral dose, except that we are not able to calculate clearance and volume of distribution after peroral dose. But we are able to calculate half-life. Cmax is more important after peroral administration, and also the AUC is something that we need for further calculations. So then let's look into some of the key parameters. Firstly, there's the AUC that I already mentioned, which is the area under the curve. And I've just highlighted that the AUC after IV injections got a different term there than the peroral. So basically, if we have an IV injection, the profile, the area is just under, of course, that curve. And we have a value for that, which was shown earlier as well. And also for peroral, we can calculate the AUC, and already just by using these two values, we can calculate the bioavailability for this compound in this species. So I've just presented the plot formula here and then inserted the values from the tables earlier, and it's dose normalized, so we have to take into account the dose. So if the doses are the same, then this part won't be needed. But for example, in this instance, the bioavailability is only 8%, which probably might seem quite small comparing these two AUCs, but we have to bear in mind that it's semi-log plot. So if I quickly go back to the linear plots, we can see that there is quite a difference in these areas, and we will have to remember also that the dose was higher in the oral group. Let's look at Cmax and Tmax. This is pretty straightforward. So Cmax is the highest concentration. It only means, actually it just means that it's the Cmax that we have observed. We don't really know if it's actually occurred before or after, but it gives a good estimate, especially looking at a plot like this. Next, we have clearance, which is an important value. It's actually not something that's intuitive. It's a value that's been invented by man. It's basically clearance is assumed to be the same for the same compound, and it's calculated by dividing the dose with the AUC. Then volume of distribution is important. That's also a value that's not based on something that actually happens in the body, but it's the mass of compound that's in the body at a given time divided by the concentration, and it gives a volume at which the compounds divided at this mass if the concentrations C to the P. Most people at least have an idea what a half-life is, so it's the concentration or the time where the concentration halves, and it's obtained by fitting a curve to the elimination phase. So firstly, pharmacokineticists or the software for that matter would have to check which time point to use. So for example, here you would start from 2 hours and then go to 8 hours. So for IV, this is what the curve fit would look like, and for per oral dose, that's what the curve fit would look like. And we would want them to be similar for this same compound as well. That's something that we like to see. All right. Of course, the profile doesn't always look like this. This is just something that I have made up, but it could be this type of idea looking as well. However, in the next slide, I've just got a real R&D data from our lab, and it shows just that the IV profile is fairly similar, but the oral profile is showing a secondary peak, and there's an explanation for that as well, which might be hepatobiliary circulation. And the values for pharmacokinetic are shown there as well. This is actually the last couple of slides. So as I've said before in this presentation, we need to have a preclinical formulation, and the purpose of the preclinical formulation is to give the compounds the best chances of actually making it into the body. And for IV dose, we need to be able to solubilize the compound, which is important. The compound cannot be given as a suspension, so it needs to be solubilized. Otherwise, it'll block the small veins in the body, and it might not be lethal, but it will show in the pharmacokinetics. For a preclinical formulation, we need to bear in mind the volumes that are allowed. So for example, for injection, we might first try and develop a formulation that can be given at 2 milliliters per kilogram, but then the concentration of the compound will need to be higher. If we use the same dose with 5 milliliters per kilogram, then the concentration is a little bit lower. The same applies for oral formulation as well. And we have to remember that the formulation should not change the properties of the compound or the behavior of the compound. And of course, it cannot be toxic. Ideally, it would be somewhere near isotonicity, and there shouldn't be much local irritation, even though even slightly lower pH can irritate the animal. I've just so basically when we start formulating, we go to these tables where common excipients have been listed for the percentage they can be around in a formulation. And there are many papers for this purpose. I've just got a few here for as an example. And well, I should say that the preclinical formulation can be fairly aggressive compared to what you can give to humans. This is a type of diagram that can be used when formulating a compound. So basically, we would start from here with the solid compound, add organic solvent like DMSO. If the compound dissolves, then we might add saline. If the compound crashes out, then we might go back to adding DMSO and repeat this process until we reach the highest concentration for DMSO. Then we may need to look into some other organic solvent like propylene glycol or polyethylene glycol. Again, if the compounds dissolve, then we go back to saline and so on. And until finally we might hopefully reach and have a completed formulation. Sometimes we don't, and then we need to look into some other options, maybe try some other excipients or even try to reduce the dose or increase the dose volume. There's really no other option if it's an IV formulation. Per oral formulation, it's of course okay to have suspension as long as the suspension's uniform so that it doesn't settle, or sometimes it might form these flakes that could be, I think, pH related, but it needs to be milky looking formulation, basically, that goes through the gavage nicely. This is the last slide. So just to cap off, animal experiments are needed and they are strictly governed and monitored, and they are conducted when there are no alternatives available. And we need to make sure that we create useful data, otherwise there's no use running these experiments. And finally, that the purpose of an in vivo experiment is to predict the pharmacokinetics of the compound in humans. So that's the purpose of the experiment. I think that was the last one. Yep. So that's it. I'm open for questions.
Thank you, Janne, for this excellent talk and this very extensive introduction to pharmacokinetics. We'll have a few questions here. Do you have experience working with stereoisomers? And can you, for example, separate enantiomers in a PK experiment?
Good question. I was talking about the column, but I didn't mention that we have chiral columns. So the column is the separation tool for the LC system. So we can have a selection of chiral columns. We can, for example, dose a racemic mixture of a racemic compound and then separate and see how they behave in an animal, if the ratios change, or we can just dose the different enantiomers and see if they convert, or study enantiomers might have different pharmacokinetics as well. This is something that we do regularly.
Okay, thank you, Janne. Another question here. How do you correct for any matrix effect?
You can't correct that by means of calculation, but you need to separate the matrix from the actual peak for the analyte. So you need to have them separate. So they need to be separated, or we need to look into purifying the matrix, whatever component we need to purify that.
Okay, yeah, thank you. Are larger animals planned for the future?
Yes. So we are planning to include rabbits. We are already in talks, and we just need to apply for a permit. I've actually got a history in rabbit studies as well, so it's not a massive issue, but we just need to find time. It's probably I think we will have rabbit studies running by the end of this year.
Okay, yeah, thank you, Janne. Further question here. How long does bioanalytical LCMS method development typically take?
Yeah, I mean, certainly when I worked during my PhD, it could take weeks and weeks, but in a professional lab like at Admescope, it probably takes about 4 hours or 6 hours.
Okay, yeah, thanks. Can you also do radio-labeled experiments?
Yes, we have permits for tritium and carbon-14, so those can be readily run, and we have done those experiments as well.
Okay.
In metabolic cages, but it doesn't have to be. It could be anything.
Can you perform bioanalysis of biologics as well?
Yeah, so we have quite a few systems. So for peptides, if they are considered biologics, we just run a normal LCMS. If it's a much larger protein, we run what is called surrogate method, and I should say I'm not the expert, but the head of biologics, I worked with him here, so I'll at least be able to describe in my own words. So basically we trypsinize break down the large protein, and we find some surrogate peptides from the protein that are specific for that protein, and we analyze them as we would any smaller compounds. Actually, that method works quite well for antibodies as well, so because they commonly have the constant, the FC regions, same for all antibodies, so we don't need to develop a different method for a different antibody. We can use the same method. But I mean, we also, in addition to that, we have different types of enrichment methods, magnetic, paramagnetic beads, and so on, ELISA methods for the biologics.
Okay, great. Yeah, thanks, Janne. I think, yeah, we're coming to the end of our time now. So I think we better wrap up. Yeah, Janne, thank you again for this excellent talk. I hope you, the audience, enjoyed it as well and learned a few things that you didn't know before. And yeah, with this, we'd like to again remind you, please follow us on LinkedIn, Samaris and/or Admescope, and watch out for further news and details on further webinars. I hope you enjoyed tonight and tune in the next time. Thanks very much. Bye-bye.